Yes, so good morning to all. So we have started to talk about certain selective methods, right? So into it, the major three methods we can go for the selection: physical, chemical, and biological. That means what we are looking to select, that is our microbes. Which kind of microbes we are looking, that is our main target. If I want to select the student, I will still have some parameters in my mind. For what purpose? Main target. I'm looking to select some students. I should have something in my mind. I'm looking for some sports. If the some sports events are there, I will look for the students having this capability, those who are good in a sports. If I want to send you in some one conference, this one is a research conference, then your research aptitude will be my important criteria. Okay. If I'm looking to send in a group discussion, okay, group activity is there. So my parameter is going to change like that. When I'm looking for the isolating the bacteria, something will be in my mind. Accordingly, I will select the methods, whether I will still go for the physical, chemical, or a biological method. Okay, so one by one, we are going to see this few points we have seen. It's a physical method. So if I'm looking for the isolating the endospores, so we may increase the temperature. Heating treatment is going to cause the bacteria, not on all bacteria, certain kind of bacteria having the capacity to develop the spore. The spore-relating bacteria we can isolate out of it. Just we have seen, then we have seen the next point. It was related to the temperature. One of the great factors. Okay, all the environment is going to change, varies with the temperature. Also, the people are currently saying about the temperature as well as the pH, as well as the geographical locations, which is not currently been included into it, but the location is the unimportant part. That means you are going in the rocky areas, or you are going to near to the wetlands are there. Or another is unimportant nowadays. The people are going to say that is a threatening area, that is the underground water. Currently, the pumping is required continuously. If you are looking to the Rajkot and nearby city, or if you are going into any of the places in the world, we are looking to use the pumps to take out the water from the below the area. Okay, the groundwater, if you are just looking, the soil is rich in the microbes. We also understand that petroleum products are being formed since centuries within the beneath to the soil. Okay, at the very depth, at the below the depth. So all the microbial community is working there. They are doing something and something. So when you are looking, when you dig up, using the some tools, and you are going to dig up to maybe it's like a 500 feet, 600 feet, 1000 feet. So you are looking to either you are going to disturb them, that microbial population is growing over there, or else you are inviting them to come outside. It is like the people are calling in the certain language that the demons are there within the, you can call in Gujarati or in the Hinduisms also are calling that Satan, Patal Lok Marej. Right? It is nothing but the water. As you are moving down, that is the rocky lands are coming, water are also coming. So all are this all of the demons are there. They are ideally sitting. They are doing their own job. But what we are currently doing, we may reach up to 2000 feet or more than that. Currently, to get the water, we go in the dig up. While digging up, we take the water. Along the water, that microbial population is coming out. And this microbial population is causing the new kinds of diseases. And I hope you may be also clear with what are the microbes, what is being included in microbes, right? Like viruses, bacteria, fungi, algae, protozoas, many things we have just yesterday discussed. I will ask that thing same maybe later. Okay, so you are inviting them to come out. Another, right now the scientific studies are telling that at the Antarctica and other places, major, we are knowing that a global warming is happening. And because of that, ice glaciers are start to get split out. The largest of the ice are there. Okay, 5 kilometer, 10 kilometer, largest, it gets splits. When it splits, it is, you can say their thickness is very high in a kilometers even from the depth and others. So since centuries, it was been like that. Air was been already packed over it. Now it has been opened up and exposed to the environment. I think earlier given the one example related to the Giza pyramids, if you do remember, right? The Kumsa was the one pharaoh whose cover was been opened up. Okay, when they have opened up by the scientists, a century back long, when they have prepared their mummified mummies and they have added some materials to store it. Some microbial populations were existing and it was converted like it is the, if you are students here are maybe a 50 or 100. Currently, maybe I'm just looking out, so 20 around. Okay, 20 students are there. If maybe a starvation is there, if you are being get locked by chance for about a 10 or 15 days or a month, everyone may have their own waters and everything. Then after, if you are having the certain materials with you, there will be a fighting. Okay, for the survivor. Then may happens one to another stage may goes on. Right. Like that only. Fungi, the bacteria and other alls start to grow on one to another. They want to survive. So they will start to release certain chemicals. Okay. Or they will start to grab it. So when it has been opened up after the centuries, it has been formed that some specific kind of a fungi in a sporulating form, they were get stored because the no much nutrient availability was left. Okay. Whole body, it was decayed. Material was there, what decayed. Okay. Now it is not utilizable by any biological community. Such product was not left. So they will start to grab one to another. It is like a cow dung. Initially, you may find that some bacterial populations is there. Above there, fungi may start. Above there, insects may start to eat the fungi. Insects being eaten up by some birds. And that birds going to get eaten by some eagles and others. When they are going to die, they are going in a soil. Then now again the saprophytic fungi and bacteria will start to degrade it. That the life cycle is going on, or the chain cycle is going on, food chain. But it was begin get halt within that. Same like that, when you are digging up to the below 2000 feet or more than that, you are inviting a monster to be get outside. Okay, so beneficial and harmful, both the microbes could be possible. Another is a gaseous problem. So geographical site, it having the much important part while isolating it. It is not been included here, but just I'm telling into additional. Then the pH. The pH is one of the important factors as well as like cell size and motility. Just I have told about two things into motility. Okay, so it was related to the physical. Now we will move for the chemical methods. So why and how we can go for the chemical methods? If you are using the nutrient broths, okay, it having the whole zone. But if you are looking, the bacteria only maximum using the carbon or the source is the nitrogen is there. If I'm looking to isolate them, what I can do? I will go for the minimal media. Do you know about the minimal media? The minimal media is having the as much as less nutrients as required. What we generally required, it is like a, if you require the one dish every day, full dish you are eating, it may having the certain breads or butter or the chapatis are there, 5 or 10. Okay, but it will be given only 2 rotis to you. Okay, maximum to it. So it will limit the growth. Now I will going to give the some specific supplements only to you, like some salad. No any other things. Everything is taken out, just only a salad is given. One day I will give for a week cucumber. Then I will give the carrot only. If you want to survive, you need to eat only. So those who can survive, okay, within that part will grow. So like that, we may limit the nutrients on it. Initially, we have isolated. I think you have done the practical job, right? You got the some contamination. Now, if you are looking or interested in certain particular kind of bacteria, you may just take a nicrom value. You may transfer to the next medium. which having the only carbon as a major source or the nitrogen as a major source. So those they can able to manage with this two or the minimal part will grow. So like that, our chemical nutrients requirement will go. It's called enrichment. That means enriching our media for one particular targeted chemical. We are not going to use the normal or nutrient broth. We may use only a particularly less required or the less amount of the chemicals will be there. So isolation of nitrogen fixing bacteria, nitrogen gas supplied as a sole nitrogen source. So you are going to give the nitrogen from the outer side. I think during the anaerobic respiration, the isolating the anaerobes, I have told about it. We have the one anaerobic chamber. So anaerobic chamber will be supplied with either carbon dioxide or a nitrogen gas. So nitrogen gas will be supplied from the outer part. So this can be easily possible. Second is like use of specific carbon sources. We may go for the some particular kind of a sugars. Okay, particular carbohydrates are there. One of the given example is related to alpha conidendrin. It's a constituent of wood part. Generally, the wood, the plants are growing in the hilly regions are having there as a part of their wood. So you may isolate it, and you are looking that only to give this as a carbon source, nothing else. So you are not going to add a glucose or any pentose containing sugars, or you may go for the others. We are not going to add. We may going to add one particular kind of sugars into it. If it is able to survive on it, we may proceed. It is like that. You may all having the like veg and non-veg preference, right? If it is a jumble, I may give the only a coconut water. You want to survive coconut water, and the coconut I will give. Okay, it is not a veg, not a non-veg. Okay, we can consider as a veg plant. Okay, but it is a like a fruits will be there. I may give only a coconut to be get it. How many days you can survive? A year, a month, or a week? It's depend. You may initially don't like it, but the coconut is considered as a full nourishment material. It's having the micro and micronutrients, and the along with it, the carpels part. There's a white color fruit is there, which is also a good source of the energy. So you can easily survive. Right. So I'm looking that can you able to grow or the maintain yourself with the coconut? You will be like that. I will restrict them. The bacteria will be restricted for one particular kind of a sugar. So sugar eaters for particular, it's only going to grow. Why we are looking, just to hear the some example is given. What is the need of certain things? Can you think? If I'm saying about this physical or the chemical, I'm looking to restrict with this. What could be the applications in the real world of such restricting things? And to grow one particular kind of a bacteria, what could be the benefits of it? Why we are looking to isolate such kind of bacteria? Yeah. Yes, I'm telling that I may use the certain specific carbon to grow such bacteria. Okay. What is the need of doing such thing? Yeah, they will, okay, they will grow. But what is the application? After isolating it, you are telling that you are going to add such sugars in media only, right? After getting this bacteria, what could be the application? Why I'm looking to isolate? I'm going to make you just on the stay on the coconut water, or the coconut will be given. Okay, green coconuts will be given every time whenever you are hungry. I will give the one or two coconuts, not nothing else. What is being a motto behind it? Okay, nutritional classification. Okay, generally you are or the bacteria were able to grow in the different also. Okay, now you are looking for only one. Okay, nutritional classification. What I will get the gain out of it? Feel it, feed it. Okay. So that is right. Okay. The way we can understand. Okay, fine. What could be the benefit I will get? Everything we are going to do for the some benefits. Okay. Classification is done. Fine. What could be the after the classification? Anybody? Okay. How we can going to use this harmful or useful? Everyone has their own thoughts. Now, I થોડું
Some
ગુજરાતીમાં
industrial
બોલો.
use.
For the industrial use, okay. What could be the way of the industrial use? Yes, you also can think. If I'm going to isolate particular bacteria which having the particular habitats or the eating habits, what could be the benefits I can get out of it? There's a sole application of the microbial. Yeah.
Bacteria, there is some enzymes, so we can extract the enzymes.
Okay, one good example that whatever as a sole carbon we have given, they have to release certain kind of enzymes or chemical to utilize it. So particular targeted pathway we can work out. Very good. For the metabolic engineering or the genetic engineering purpose, we may go. Very good. Okay. Another thing. Pardon? Yeah. I couldn't understand you.
Some of the bacteria is harmful, infectious. Dries and chemical dries.
Yeah, that is also good. That from the mixed populations, this isolation is particularly to get the that bacteria or the microbes out of the whole population. Okay, and which may be beneficial or the harmful for me, right? So I'm looking for it. That if it is the harmful, I'm looking to control to use some particular kind of antibiotics or the chemicals to control it. It may be going to spread the certain kind of infections or the pandemic. Okay, the possibility will be there. So that is our main target. So to get the isolate in a pure form, main target is what? To understand there either it's a beneficial or the harmful. Main target for the every pure culture first. If it is a beneficial, we will go for their applications in our industries. If it is harmful, we are going to use the certain applications already existing in the industrial part to control it. Okay, if it is causing the certain disease, not only the human, maybe in the plant or animals, they are going to cause the certain, the animal husbandry. If the cows or the any animals, camels are get infected, they may get died. Okay, so how we can save them? So we need to develop some antidotes. So first to do any sort of the experimentations, we need to or the must be in a pure form. That is a definition of like a pure culture. So application of pure culture. So how to get that pure culture or selections that I'm just discussing. So initially on the first day I have talked with the pure culture. Since long our talk is going only related to pure culture on. How to get that pure culture. The application of pure cultures are applied over here. So how to get it? We have seen is the anaerobics or the aerobes microbes where we have seen. Now we are looking for some selective ways, physical, chemical, or the biological means to isolate in a form of pure form. So if I am restricting, I got come to know that this only sugar is being utilized. Why I have selected? There may be a certain reason. Suppose you may think like that. We know some example we may take of species. Some specific species are having suppose a very worth value. If it is a one species are there, 1 kg may be sold in the 5 lakhs rupees. Just think light in broad. They may start to get some infections in particular area. Okay. And when they have dissected, they come to know some bacterial populations is there. It having the certain normal flora and some bacteria, maybe a mixture is there. So how I make isolate them first? So I may think that this fish is continuously feeding on some particular kind of a plants or animals, and it has rich source of particular some sugar only. It is infecting in digestive tract. Digestive tract is full with the some amino acid or protein or the sugar. Okay, if I want to isolate one particular poisonous or the infecting bacteria, okay, so I may assume that it may consume my given or the given sugar available in their digestive tract of that fish. Okay, so what I can do, just I will going to dissect that fish. I will take the some whole the microbial populations out of it and try to grow them in the artificial media, which is rich in the one particular sugar. Okay, so this is my targeted work. Okay, so where there I will make able to grow some pure cultures. Some other normal flora I will going to put it out, but the selective I can find it out. It is like that. You may be a total 100, right? I may make a certain conditions for the selections based on the food habits. It may be able to eat veg, non-veg, chili, okay, and the sour and the in a hot condition. How many persons is going to fill with? It's going to reduce down, reduce down, reduce down, and I may add the one another parameter at a night 3 o'clock. It able to at least eat 500 gram of this given amount. Okay, among them also, if the 10 students will be there, it's going to reduce to 1 or 2. I have gone for that selections to get the pure person who is fitting in my criteria. Okay, so that's I'm looking in this pure culture identification. One particular targeted part I'm looking. Now I'm going to culture it for a larger. It may be benefiting, sometimes happens. That fish, when it is having the higher amount of the bacterial populations in their tummy, okay, they will start to grow very high. So I'm looking to overgrow them, otherwise to control them. So beneficial or harmful, that I will decide later stage. So this is my main motto. I hope it is understood why we are looking for the pure culture. Sometimes the dilute media, yes, that bacterial populations, that's what I have told, that you are having the habitude of the eating the 10 breads. I will give only 2 breads. Okay, those who are the hungry, very high, will not suit in my suitcase or in my suit. So they will move out. Those will fit in my shoe will be there with me. Okay, so it is the basic concept for the dilute media can be used in these processes. Okay, another is the inhibitory or the toxic chemical. That means I'm going to add one particular kind of antibiotics which is going to control all the gram-positive bacteria. There will be no growth. Okay, another antibiotics I may add which only allow the bacillus to be get grown or the cocci-shaped organism is only able to grow. Okay, so it means or we may arrest their protein synthesis. I may add some particular antibiotics which going to control the gram-negative and their particular parts, or some particular kind of microbes can be grown. So I made the certain toxic chemicals or antibiotics to control one population. It is like that out of hundreds. I may give some chemicals into it. It is like the suppose it's like a some particular drugs. Everyone cannot be easily able to take it. Okay, but others can. They will like to enjoy it even because it was liked by them by chance. Or it's like alcohol. It's a dry state even, but everyone is not like to have the alcohols. Maybe it is in little amount. If it is given that in my conditions, every day that students has to consume 1 liter, it will be very high. But if it is able to manage, okay, then it will be accepted. So it is like toxic. The level is going to be a very much toxic. The other is going to get nullified by itself. None of them will come. They will say about like a 250 ml. If it is allowed, we may come. No, condition is 1 liter. Okay, so those able to get that 1 liter and able to manage will accept it. So here it's the same. Toxic compound is given, other bacteria is going to die. Those able to tolerate the toxic compound are accepted. Okay, that means on the next step, I came to know that if I want to control this bacteria, I need to increase or change my antibiotic because their 1 liter capacity is daily. I have isolated that guy at having the 1 liter capacity for the liquor. How to control that, guys? Okay, so I need to control in the different parameters. That persons cannot be controlled with the 1 liter. But having the daily habituous. So like that, we may go for the isolation of bacteria, which only allow growth of the one particular kind of organism only, not all. Okay, those able to survive, then I also come to know that it is their basic habits now. If I want to control them, I need to change their another parameters. Understood the concept? Why we are going for the different things and the chemical also? Getting? Fine. Let's move ahead. Yeah, here can be intuitivity, sometimes the heavy metals even. Just the heavy metals, antibiotics, or there's some culture conditions, salts. Salts also are sometimes toxic. It is like a fresh water and the marine water. If you are isolating the bacteria from the fresh water, they cannot tolerate higher salt. They will die. It is like that. If I'm going to give the 1 liter, okay, water will be given. It is highly salty. Or I will give the daily for a week water sample, highly sure. It is like a lemon juice or the lemon is totally is given to you. How you will survive? It will be difficult. Okay, another portions with the sea water is generally given. Those who are in the acquainted with the fresh water will not able to survive in this salt condition. So it will be going to make them problem. So you may go for some innovative methods. You have to. You may use some solar systems or the furnace is there. You may boil it. Distillation is done. You may get your water, but during that part, out of 1 liter, half liter may gone up. Okay, by a steam in processing. So you are also then are accepting that at least I'm getting the half liter for my day. So you are able to manage. Those able to manage are being accepted. Okay, just the targeted part is what? Those who can either salts or salts, bile salts, others. There may be two things, tolerant and resistance. Okay, what are the basic difference between tolerance and resistance? Can you say? There is a three major terminology otherwise, sensitive, tolerance, and resistance. Can you? Very good. Okay. Okay, then sensitive. First was right, second was doing some error. You may going in a third step. Okay. Which error? Huh? You are mixing the first and second options now. Okay. You are near to, but you have start your yes, sensitive, resistance, and tolerance. You may just think again.
Sounds like you're not taking the chemical of the
Okay.
but tolerance you take, but you get the lots of it.
Okay. Then sensitive. Okay. So three things is there. Sensitive. It is totally get avoided. It is like touch me not plant. If you just touch, it's the sense highly sensitive. It's going to close it. With a strict non-veg and strict vegetarian. Something is given in their plates, they will not able to accept it. Highly sensitive. Okay, about it. then the tolerance up to certain level they can tolerate. Okay, up to certain level they will tolerate. Resistance, nothing is going to happen. They can resist fully. Whatever you will give, veg, non-veg, or the diseases are there, they never bother. They like to eat, they will eat, they will never not. Tolerance means up to certain level they will tolerate. Afterward, they will reject it. Okay, so in this case also, when you increase or decrease the amount of the metals or the heavy metals, okay, heavy metals, some bacteria are highly sensitive. Just little amount is there, heavy metals, they will die. Okay, now I increased, like it is a 0.001 millimolar concentration was there. Now I have increased to 0.1 millimolar. So they will tolerate. I may go up to a 1 millimolar, they will tolerate. If I increase to 10 millimolar, that bacteria may die. Okay, so I will go in selections. Now I increase from 1 millimolar, 10 millimolar, I may go for the 100 millimolar, thousands millimolar. Then also that bacteria will injure. Nothing is going to happen. It's resist to it. Okay, it can resist anything. That bacteria I'm looking or what? So the selections based on the concentration is also going on. Like a salt. Some bacteria may require minimum amount. Okay, it is like a 0.01 millimolar. But I may increase the concentration, then they are going to get sensitive. Minimum amount is okay, but after it's a problem. It's like a if you're adding some salts in your dishes. By chance it has been added twice. You may think, okay, I will mix it well because I'm hungry, I will eat. But there is a 5 times it has been added. You will not like it. You are going to be considered sensitive. But even after, if you're hungry, 5 times salts is been added, you are going to eat it. That means tolerance. Okay, you are tolerating it. But other thing is like that. It is like a cake is there. It should be added with the sugar. It has been added with the salt. And you are eating. One has to. Right? In that context, you are resistant. Nothing is there. I just looking to it. So I made. Okay. So it is a resistance power. It is like a parrot. It can eat any sort of the mirchi. We know that red hot chili is there. It can eat because their sensitive birds are not there. So it can eat. There is a resistance. Okay, it having the God gifted or natural resistance is there. If your salt sensitive birds are being get inactivated by any of the process, you never come to know that I'm eating the salt. May happens if the certain chemicals I may give you for the drinking on a day, for at least 1 week, your all the sensitivity related to your tongue will be lost. Then after I may give anything to you, it is a cake fully with the sugar transferred in a form of a salt, you will like it, you will enjoy because you don't know what I'm eating because the sensitivity is lost. So resistance may become the natural or sometimes induced. Okay, the induced resistance also come or the induced tolerance may also get acquired. So here we are not looking in that part of the research, but we are just talking that what could be the applicability. If I'm going to add a certain dyes, okay, if I'm going to add the particular dye into the one nutrient broth itself also, okay, and broth I may prepare, I may add some particular dye. The bacteria will not grow, or they may get sensitivity. They may able to tolerate. So they are not going to do anything with that dye, but they can tolerate. But sometimes the resistance, nothing has happened, or they may start to utilize it also as their energy source. And whatever it's been given, it's like one of Pseudomonas, very notorious organism in the world, very notorious. It can survive in any damn condition. Many species are there. Okay, they can survive in any damn conditions. You may give the heavy metals, you may give the dyes, you may give the petroleum products, you may give the acid directly, you may give the alkaline conditions, anything. Okay, it is like a guys in militants. Okay, they are like a in a form of a terrorist or a soldiers. They have to get trained in that condition. They able to survive. Like Sometimes you may see have seen the some in a Discovery channels. Mr. Bear is coming. Uh, One of the program is coming, right? Survival is the fittest, like a thing. He is going in the mountains, in rivers, and so many areas in the world, and he able to manage that how to come out of it. Team members are there. He may eat just any insect. He may eat any fruits. He may eat any leaves, any vegetables. He also able to survive on the snacks. He may use his own urine to hydrate himself. So the way he manage himself, it is totally different. Okay, it having the capacity to resistance to do any sort of the things. So it's kindly totally notorious, totally different kind of things. Okay, the person is totally different. It's not a notorious, but known for its good things, for into that category. Okay, so such thing is can be possible. So we are looking for the either tolerant, resistance, or a sensitive organism to get isolated. So when you are going to add one particular kind of a salt, maximum population is going to get sensitive, will not grow. Okay, in my 5 conditions, what I have told earlier, non-veg, heavy chilies, this is the 500 gram at a night 3 o'clock and in hot condition. It should be served and within 30 minutes, one has to complete. Okay, so how many will fit into it? Very less. All are going to get sensitive and tolerant, maximum. Okay, and it will be given freely every day. Selection is also there. Okay, but how many will fit into it? Very few, very less. I may change the certain parameters now. Okay, it will be sweet dish, 500 gram, highly chilled. Okay, given with the one particular cold drinks of their selections at 2 o'clock around noon. Okay, or 12 noon. So many people will fit into it. Okay, so this is the with our given time period and 30 minutes will be given to you. So at least few people will come to try over it. So there is a tolerance will come. I may change now menu. Okay, I may change. It is at around 5 o'clock. First, I'm telling the time now. I may add the like either it's the choice will be given is a sandwich or the pani puri. Okay, third will be amount will be the 3 dishes compulsory. Okay, within 30 minutes you have to complete it, right? And it will be supplied along your required chutneys. So how many people will fit into it? So many will fit into it. The reason is what? It is the liked items. Okay, so it is not required in the tolerance and they are already fit into that part. It is their normal diets. It's like a thing. So and their like dishes are there. So they all were coming into the sensitive. They converted into resistance now because it is the normal. But I may add now another parameter into it. Okay, that dishes will be served without chutney or it having the red hot chili. No, 2 conditions only. Okay, so coming in the picture, people may shift into tolerance or in a resistance. Those who are able to eat without chutney, they will come, or with hot chili, they will come. So there is a changes are there. This all sort of the changes are playing while in microbiology. That's why I'm telling those who are good in the cooking or in the food related things are good in a microbiology because we need to understand their phenomena and phenomena physiology. What we like, how we like, in a way or format, same thing is happen with them because they are the living entity. All are living organisms. They all can sense every sort of the things. What we sense, they also sense. So if you are good in the cooking, understanding of the food and the cooking, okay, eating habits for the one to another, then you can be a good in the microbiome. How to do isolations? What need to do after the isolations? Okay, like preservation techniques. After this topic, I will take you into that part. Like you may prepare the certain foods, or the certain items are prepared, like pickles. बराबर છે. Because have been prepared, we stored it for a longer of a years, maybe for a 6 months to 1 year. But same item may be prepared for a 1 day or the 2 days. The process and methodology may change, right? So what sort of the food you are preparing, that is for the 1 week to be stored or for at least for the half day. Accordingly, we are going to change our ingredients like that. What you are looking for the microbes, we need to give the some eating parts them in different formats. Okay, so I hope that major part is clear. The same thing is written here. Yeah, some example related to inhibitory or toxic chemical is there. Like a Campylobacter jejuni causes the diarrhea in humans. Two samples contain other bacteria as well. So addition of antibiotic like vancomycin or polymyxins does not allow the growth of other organism, will allow only for the C. jejuni. It is for the diarrhea. I'm looking to isolate it. One particular bacteria is there. Then from the stool sample, if I'm going to grow on the plates, okay, other microbes will start to grow. I'm looking for my own interested bacteria. So what I can do, I may add some particular kind of antibiotics, does not allow the growth of others. Only for this, because this one is resistant to it. This bacteria is resistant to the vancomycin, not other at all. Okay, so if I want to treat that patient, I cannot give the vancomycin. If it is the vancomycin is given, nothing is going to happen. They will like it as a pani puri or sandwich. Okay, it is their normal. Okay, they will be happy. So we need to go for the another antibiotics. But to first step to isolate that bacteria, we may go for the using particular kind of a antibiotics. So other will come. Okay. So what will be my first criteria from the filter it out will be a different then. Okay. I may just change the time 12 noon to 12 noon. It is evening night. How many will come for the eating the sandwich? Maybe a 50% may reduce, 70% may reduce, because at 12 you may be at your home. Why to come here? Yes. So your first criteria with the geographical locations, everything is for change. Right. Just one parameter, if you play, you can get the many things out of it. Okay, now there's some animals method. Okay, till this, anybody having any question? It's a 9:20. I'm not going to take more, otherwise birthday zone.
Because bacteria in media, how much would stay in the bacteria?
It's depend upon the kind of a bacteria. Each organism having that's we are going to discuss more in the growth curve next unit. Each bacteria do have their different method of a life cycles. Okay, some are the slow grower, some are the fast grower. They may be covers your entire plate within maybe a 24 hours. Some are the very slow runner. They will grow gradually, right? And some are running in the mediocre level. So they may wait for a while, they may go and grow. So some bacteria are there, they take at least around 1 week to grow first. You have to keep for at least for the 1 week, then they may start to grow and then may pick up in the leg to log phase. So it is called the leg phase, log phase. Exponential phase where they start to divide. It is like in a class also. Some students required to ask many a time. Okay, once they started, we need to say them to stop it. Okay, so once ignition has came, they start, but they may have taken the months to start. Some have start to bluff everything, whatever it is there in any class may. Some are the choosy. While discussion or the with which faculty they may start to say, otherwise not. Okay, some are knowing answer, but they will never respond. Okay, some are able to know nothing, not knowing the subject lectures, they will start to ask many things or respond many things. So like this only, or bacteria, we don't know when they will start to grow. So we need to wait and watch. Okay, it is like a growing certain plants. We know that every plant's having the different habitats and they're growing. If you are going to put the seeds, it may take at least 1 week or the 4 days or 3 days to sprout even. If it is like a mung only, within the next day it gets sprouted. So it's depend. Same like the plant is there, even the animals or the human are there, same it is for the microbes. When they will got ignited or the start the division. It depends upon the cell type.
If this bacteria is not safe, is more
Yeah, it's based on the sample from where you are looking to do isolate and which microbes you are looking to isolate. So we need to go for the different flex preparation. We cannot grow with a nutrient growth. We need to prepare the different flex or the different solutions we need to prepare. So it is continuously researching. Okay, the research will be required. If I'm going to look some bright student out of 50 students, I want to select at least 1 or 2 students. I may have my certain parameters. Okay, initially I assume all are equal. Okay, then I may set the certain 5 parameters. It's not always that those who are good top in marks are good. What is my requirement? Either it is a research or what? Those who are good in the plating, I'm looking for it. If they're good in the 100 out of 90, sorry, 90 out of 100 scored, but when the plate is given 100% contamination, that guy is no use for me. I'm looking for this. But the same guy is useful for me when I want to send them into some debates. Okay, those who are good in the practical sessions is scoring the 60%, never been having any contamination, but I cannot stand in the debate part. So the parameters and motto is changed like that. When the bacteria we are looking, we need to set some scale of the parameters. Then we may go for the selection. Okay, so I hope it's maybe clear. I'm ending here. You may put your attendance over it. And within the lecture time, I am going to take that response. If you are going to put this response maybe after half an hour or 1 hour, that I'm not going to consider. That means those who are present either in online and offline mode during the lecture time periods are being taken for their note. I have seen that many guys still putting their attendance at a night 9 o'clock. Okay, they recall. Okay, I have to put, but it is of no use. Okay, so within time period, within hour, you are having the Google Classroom link over for this, as well as I'm putting into here. Okay, 5 or 10 minutes, it's okay. Sometimes you are putting the network has not came. It is like a 8:30 to 9:30 lectures. You are putting at a 12 o'clock during your maybe your break time or somewhere. It is not accepted. Okay, response will be there. Google that form will going to accept it, but I'm not going to accept. I'm going to filter with your time. Okay. So I'm ending here. Okay, tomorrow we are having the one lecture, right? So we'll meet tomorrow.